presentation. Yeah. So we have Dr. Saurabh Raghuvanshi. He is a renowned computational biologist from India. He is currently the lead in Indian Biological Data Center, Regional Center for Biotechnology, Faridabad, Haryana. He is a full-time professor in the Department of Plant Molecular Biology, University of Delhi, South Campus. He is well known for his outstanding contribution in the field of genomics and bioinformatics. His group is working on developing novel data biocuration and digitization methods that would enable complete digitization of published experimental data sets. Such digitized data sets can be efficiently searched and seamlessly integrated. This would also enable development of data schemas that can be used to publish experimental data sets within publication in a computer-readable format. His research group is also working in collaboration with AIMS and Bioheaven 360 Genotech for extending the data digitization methods on biomedical sciences and developing artificial intelligence-based diagnostic web portals. They are also working on providing IT-based solutions to Indian farmers in order to ensure societal benefits of the IT infrastructure developed at UDSD. He has 16 years of teaching experience. He is also the recipient of NSI Young Scientist Platinum Jubilee Award in Plant Sciences in 2006. And today he'll be talking about the Indian Biological Data Center. The Indian Biological Data Center is the first national repository for life science data in India. And this is mandated to archive all life science data generated from publicly funded research in India. The data center is supported by Government of India through the Department of Biotechnology, DBT. It is being established at the Regional Center of Biotechnology, Faridabad, where Sir is currently working as the lead. So with this brief introduction, I would request Sir to kindly enlighten us with his work and the Indian Data Center, where he's taking the charge, and he's going to talk more about that. Thank you, Sir. Thank you for being here.
Thank you, Dr. Annie, for a wonderful introduction. And let me just share my screen and then we'll start. Okay, hopefully you can see the screen now.
Yes, sir. It's visible to us. Yeah, but it's not a presentation.
Okay.
Thank you, sir. Yeah, it's fine.
So I would start by thanking the organizers for organizing this meeting, and specifically Dr. Sumer for giving this opportunity to discuss. Now, as Dr. Annie mentioned, there's several things which I'm involved in, but this is something that I have been working on for the past year now, since last April, actually. And sorry. Yeah. So this is something very exciting and new for all of us, not just for me and my group, but at national level, it is something of immense importance, and it addresses an issue that was actually needed to be addressed a long time back, but now this has been done. That is to develop a national repository where all life science data, biological data, which is generated in the country can be actually archived. Now, if we just give it a thought, before anything of this sort was developed, all our data sets were getting submitted at international repositories. And India actually didn't have any data repository, to the extent that if you ask scientists of any branch that what is the amount of data that we have at national level, we didn't have a proper figure for that, because we didn't have a repository, and that was a big lacuna in our capacity. And as India moves towards a dream of being a developed country, you can't have that level of standing if you can't handle your own data sets. So these were the thoughts with which we have been trying to set up this data center. Personally, I have been after this idea for the past almost 10 years now. And fortunately, we are at a point where we are able to realize this aspect. Now, before I go into the details of this data center and what are its activities, I would definitely take some time in actually trying to understand why do we need a national data center? Because this is a question that comes very often to me, that when there are international data centers, why do we need to invest in India? So this is something that I would like to address through this talk today. Now, one of the things with data is that it is being generated at a very fast rate. And the term like big data, they are being mentioned a lot. Now, the thing about data is that there's a difference between data and knowledge. So what understanding you develop from the data, that is not a given all the time. That means you need to do a lot of work on the data to generate what is known as knowledge. And this phenomena seems easy, but actually it is very, very complicated. And one of the most important aspects, what we all of us need to understand, is that everybody looks at the data. Even the person or the group who have generated the data will look at that data only from a certain perspective. They are not going to look at everything that data is saying. So they would look at the data, they will do something, they will work on their own system, and then they will interpret something out of it. But that's not the complete thing that data is saying. Now, who is going to do that? That basically means that you need to share this data to someone else who is working on related things, but on a different aspect. So this aspect of sharing is very important if you actually need to exploit the full potential what a particular data set or a group of data are telling you about. And we take the example of human genome data. You can look up on the net the amount of impact it has on biomedical sciences. And the only reason why it was there, because people who had done genome sequencing of the human data shared it as an open access data. And labs across the world actually were able to access it. This is what is summarized over here when we say FAIR principles, FAIR, FAIR principles. So this journey of the data to the knowledge actually can only be enabled if we are following this FAIR principle, which basically is in kind of an abbreviation for F is for findable, A accessible, I interoperable, and reusable. That means, first of all, we need to make the data findable. Findable in the sense, I have generated data, I know where to find it. But to a person who does not know me, even to that person, this data should be available. So that is findability. Then accessibility, that means, although you can find the data, but are you able to access or download it? That is accessibility. Interoperability basically means that for any organism, you have data sets coming from different technologies. You have RNA-seq data, you have genome sequencing data, you have chip sequencing data, you have microarray data. You have a lot of hundreds and hundreds of different kind of techniques in which data, but ultimately they are all talking about the same organism. If you are just talking about a single organism, they are all talking about a single organism. Rather, even they are talking about a single cell of an organism. Now, in order to interpret everything out of it, that data sets have to be interoperable. They need to connect and talk to each other, and then you can make an interpretation. And then the last thing is reusability. Reusability is important because, for example, we generated some kind of a data today, and based on the current knowledge, we interpreted it. Now, let us say 5 years later, 10 years later, more knowledge in the surrounding areas would come up. Then it is very, very useful to pick up a data from an earlier state and then reanalyze it. That gives more knowledge generation. So these are the FAIR principles. And as a nation, we need to have the capacity to implement this kind of a thing to enable all labs in India where they can have a fair access to the data that has been generated. And I'll give an example of how we have done that. So this is one aspect, is to implement how sharing and interoperability and accessibility of the data at national level needs to be done. And why at national level? Another very important aspect is that there are several important aspects, important problems that needs to be worked out at national level only. And why only national? Because at international level, those problems are not very important. They are kind of limited to Indian demography or Indian ecosystems or whatever you can think of. So we need research groups to work specifically on problems that are related to India. And even if globally, it may not be very appealing. So we need to develop an in-house machinery, a robust machinery to do that kind of an analysis. So that's one aspect. Now, another very important aspect is that this is a term which I want to introduce to all the biotechnologists today, that is DSI, that's digital sequence information. And the issue is that this digital sequence information is linked now to access and benefit sharing. Now, what it basically means is that there is some product development or anything that could be commercialized from this digital sequence information, and benefits of this commercialization of the sequences has to go back to the people who have actually either generated the data or who have been stakeholders of that data. And these includes knowledge aspects like, especially in India, like Ayurveda and related knowledge. Then we have lots of varieties. We have lots of big germplasm, which is there for almost all crops, their information, and then naturally occurring drug molecules, a whole lot of that. Whenever these things are getting commercialized, that benefit sharing needs to go back. So this is something I'd like to have some discussion now. Now, when we talk about DSI, this is an issue that is coming up at global levels now. And at India also, there are some discussion forums, but not very, very widely spread. So that is one aspect why I want to discuss it today. Now, digital sequence information is actually, interestingly, for a molecular biologist, it may just, the first thing that comes to the mind is that it's a DNA sequence. But that's not true. Although this term was formulated in some meetings of the United Nations, digital sequence information, it comes out that there is no definite definition for that. And there are certain options in which you can actually define. So it has become a placeholder. Because you can, there's one option where you can, in group 1, you can say that it's a DNA and RNA sequence. Then you can say DNA, RNA plus proteins and epigenetic modifications, and then metabolites and macromolecules. Now, why this kind of a discussion is coming up is because when we are trying to, when these discussions move from scientific forum to administrative or legal forums, their level of understanding is very different from what we would say. So they would say, to the extent that digital sequence, it doesn't say a genomic sequence, it says digital sequence. So sequence could be a sequence of nucleotides, a sequence of amino acids, even a sequence of carbon atoms or something that's a metabolite or something. So there is actually an international forums are discussing at the United Nations how to define this term in an appropriate fashion. Now, so what the important point to understand over here is that what is this issue about access and benefit sharing on DSI? And this is what I will discuss today in a next couple of slides. So the story starts with one aspect, that was Convention on Biological Diversity. This was an international kind of a consortium of United Nations of several countries, 196 countries across the globe. And what they wanted to do was, the primary idea was that there must be some defined efforts for conservation of the biodiversity, because as humans develop, there is an impact on the ecosystem. So that conservation of the biodiversity is very important. And it was accepted that there will be a use of the biodiversity, but that use has to be in a sustainable format where the biodiversity is conserved, although it is being used for some beneficial purposes. And to draw and to guide and the base to give energy for both of these activities, there has to be a very defined access to genetic resources. That is, whatever genetic resources were used, whatever benefits were coming out of it, should be equally shared with the parties which are involved. Now, parties here would be, for example, here parties are like countries like India has a lot of Ayurvedic knowledge over here. Herbal formulations are there. So there are two things. One is the tissue from which it is being developed, and then the information. So this Convention on Biological Diversity actually was to enforce these three objectives, and it was this term that was defined, access to genetic resources, that has created a problem. Now, what the problem is, when you look at the definition of genetic resources, it says it's a genetic material, and genetic material is defined as any material of the plant, animal, or microbial thing. Now, what is being done over here right now is that this genetic material term is being interpreted where it is including the DNA sequence also. That means whatever genetic information or genetic material is there, it also covers the constituent DNA sequence and protein sequence also. So here's the dichotomy of thoughts. The Convention on Biological Diversity was actually formulated to control movement of live tissues. That is, if you have to move a live tissue from one country, or if you are making a formulation from a live tissue, then you have to have some access and benefit sharing. Not on the information, because DNA sequence information is a digital information. It's a digital information. It's not a tissue. But that is where now the people are saying that because the information itself is important, and you don't need a tissue right now to commercialize. So whatever restrictions or obligations were made on genetic material should also be done for the DSI, that's the digital sequence information. And this is why it is being done. Sometimes it sounds absurd that why are we talking about a DNA sequence over here when it was just about the tissue. The reason is this, that earlier, whenever there was usage of a tissue or knowledge associated with the tissue, it was controlled by the CBD, and there were some regulations that if there is a commercialization, you will actually transfer the benefit back to the people where you got the tissue or where you got the information. But with the open access nature of the DNA sequence data, you don't need this tissue. So all you need is that you just sequence it, you take it from NCBI, commercialize it, and then nothing goes back to this country or to this village or to this city or whatever be the dimension of operation. This is why this discussion is coming, that now, since everything is getting sequenced, we need to put some thought about the nature of the open access of data. So there are thoughts that a lot of data may not remain open access just to prevent this leak of benefits. Now, this was actually accepted as a conflict of idea at the conference of the parties to the CBD, that is Convention on Biological Diversity, and the Nagoya Protocol, where the first, in 2016, this term was introduced, and in 2018, this United Nations, they agreed that there is a divergence of issue, of thoughts on different countries of how this DSI and the related access and benefit should be there. So this is an identified conflict now, and India is very, very special in that, because it has huge biodiversity. There are 3 biodiversity hotspots in India, and as well as ethnic human population. So when we talk about biodiversity, most of the time it will not include human populations. It will include basically plant and animals which are otherwise either they are wild or they are not agriculturally important. So that's the basic concept of biodiversity here. Now, the contest over here now becomes is that, okay, while there is a debate on whether the DSI or the digital sequence information or the use of the digital sequence information should remain open access like it is these days. You can download anything from internet or from NCBI. And also, one needs to ensure that whatever benefits are coming from the use of this DSI should be actually shared with the concerned parties. So this is the debate, and this is an ongoing debate. I mean, nobody has taken a call right now, but this is a debate. And since it has been identified as an issue, so people at global level are actually trying to figure out what should be the formula. Now, but the writing on the wall is very, very clear over here that we need more scientists to engage in this kind of a discussion. And there is going to be an evidence-based compromise. Now, this is an important aspect. Evidence-based compromise basically means a compromise between these two. So while you cannot say that everything is going to be open access, you will have to take into account that you will have to find out a mechanism where you can share the benefits from the DSI. So this is the compromise. And the problem over here for the scientists is that most of the scientists are not aware of this. But if any ruling comes out in this, that you cannot share now the digital data freely, that's going to have a very huge impact on the science at global level. So there are a lot of discussions, and a lot of data sharing models have been created. Today, I won't go into the detail of these models, maybe some other time. I just wanted to show you these. So these are all different models which have been proposed. And model number 1 is basically when you are saying that DSI or digital sequence information should be treated as equal to the actual tissue or the genetic resource. And here, so that's model number 1. So that's a most strict one on use of the digital sequence information. And then there is something where there is no benefit sharing from the DSI. And then these models are somewhere in between these two aspects. So where do India stand? So India also has made Biodiversity Act in either 2002. And as per this, India has recognized that it interprets DSI, that is the digital sequence information, as an integral component of the entity genetic resource. So India has already accepted this phenomena because India is housing a lot of traditional knowledge on Ayurvedic and other plant-based and even animal-based products. So they have already accepted this fact that DSI is part of genetic resource, and DSI, that is the digital sequence information, cannot remain, the access to it cannot remain totally free. There has to be some restriction on this aspect. Now, seeing this phenomena, in the year 2021, Department of Biotechnology came up with the Biotech Pride Guidelines for promotion and exchange of research data. Now, these guidelines, the basic guiding principle is that life science data from India should be treated as a national heritage, and it is with international implications, simply because this knowledge is very, very important, and everybody in the world would like to use this knowledge to benefit their own, to benefit mankind in general, or to benefit their businesses also. So we need to, it must be conserved within the nation, first of all. If you have to figure out a mechanism where you can share the data, you need to have the data, only then you can actually regulate it. So consolidation of the data is very important. Accessibility of this data to all national labs. So what Now, this is a very simple statement, but what it is important one, because if you look at current scenario, if I generate that data, and if you have to use it, you will have to go to an international server and then take it. You won't be able to find it somewhere, anywhere in the Indian servers. So that is what it means, accessibility. And ensure maximum knowledge generation, and critical for long-term self-sufficiency in R&D, because this is a very important thing, because unless and until as a nation, we are able to handle the amount of data, and I'm talking about petabytes of data. So petabytes, it's 1,000 terabytes, and 1,000 terabytes, and terabytes is 1,000 gigabytes. So it's a huge amount of data. And when you start to handle that kind of a data, it's a different science, actually. That level of expertise in life science needs to be generated in national level. So with this, the policy was drafted, and it was actually alarming to realize that there is a National Data Sharing and Accessibility Policy, NDSAP 2012, but this policy does not have any special guidelines for dealing with the biological data set. That's why Biotech Pride guidelines had come up. And all these departments, including National NBA, National Biodiversity Authority, Niti Aayog, Ministry of Home Affairs, Department of Biotechnology, DST, and all they were consulted to formulate this guidelines. And also the international guidelines which are applicable over here, including the Nagoya Protocol, they were all actually assimilated over here in these guidelines. Now there are you can download this from the net, and I'll show you where you can get that. There are two important things. What these guidelines say, there are two phenomena when you talk about data archiving. One is data submission and data access. Data submission to a repository does not simply mean that the data will become accessible to everyone. You can actually control this stage. That is, you can submit the data and you can control when it will be accessible to others. And this is very, because you can imagine that working as a research scholar, working as a postdoc, you generated some data, you would like some time to, you would like to analyze that data yourself before giving it to others. So that's understandable. And that's why data submission is one aspect. You can hold it, and data access comes into picture. So these guidelines, actually, they basically have, it ensures three dimensions of data access. That is, one, definitely there will be an open access data, but there will also be data sets which are under managed access. Managed access basically means not available to everyone, but once you sign an MAT or something, that data will be made available. It could be free of cost or whatever. And then there could be some data which is totally non-accessible, and these are from national security point of view. For example, human data from armed forces, it's a no-access data. You can't access it, but it is generated. So that's a no-access data. And it covers everything, all kind of data sets which can be generated. This is not an exhaustive list. Any data set that you can imagine generated comes under this Biotech Pride guidelines. And in order to implement these biotech guidelines, these data submission guidelines, the Indian Biological Data Center was conceived, where the basic idea of this data center is that all life science data generated across India will be archived over here. It will be curated. That means more information would be added to it, and then it would be shared within India as well as the globe. Now, about this data center. Now, this data center is primarily housed in Faridabad as Digital Center of Biotechnology. This is where the entire data portals and data submission processes have been developed over here in Delhi, in Faridabad. And because this data is of immense importance, there is a disaster recovery site at NIC Bhubaneswar, at the other side of India. So there is a real-time data exchange between these two. And the basic idea is if something goes wrong, we can retrieve the data from, we can retrieve the data from here. And in Faridabad also, in the RCB IBDC data center, here also at least 3 copies of the data sets are made, so that if there is any breach of security, the data would be safe. Now, I'm pleased to say that we had started work last April, and within a year's time, we have been able to come at a stage where the data center is operational, and the minister was able to inaugurate it last month, and it was widely covered in different news items. Now, this is the data center building over here. This is the insides of the data center. This is the Brahm HPC, that is the high performance computing, and the data storage over here. The specifications of this data center is that a combined data storage of about 5 petabytes, that is 4.5 petabyte is at Faridabad, 1 petabyte storage space is at NIC Bhubaneswar. And so a combined of about 5 petabytes of data can be stored in the data center. And this will be increased as and when there is a requirement. And there's a huge compute capacity also. And in all, about almost 900 teraflops of compute capacity is there at the data center. Now, what are the basic philosophy for developing this data center? Now, because the guidelines say that data center has to take all different kind of data sets, different kind of biological data sets. So there is no way you can have a single portal which will take up all kind of data sets. So IBDC is being developed in a modular fashion, where based on the data type, there are different data portals are being developed for different kind of data types. And then there is a security check for those portals, and then the usual process happens. One of the very important aspects for the data center is that it is not conceived or not imagined to be a center in isolation. The first thing that we did was to ensure that within its core, we develop a thought process that there has to be an extensive level of collaboration in different dimensions with national as well as international levels. So we have already identified several levels of collaboration from 1 to 4. In 1, we are collaborating with international repositories, and then with ICL2, we are collaborating with national level institutes, as well as in ICL3, major projects which are generating data also. And so in the ICL4, we are collaborating with groups which have a very extensive knowledge in their field, and they would help us in developing the curated data sets. So we'll provide the backend facility, they will provide the scientific expertise, and then develop. This is the website, this is the data portal. You can actually visit ibdc.rcb.res.in, and you'll come up to this web page, and you can want to submit the data. You can click on this button, and if you want to take a look at the tutorials, you can look at on this tab. This is a copy of the Biotech Pride Guidelines. This can be downloaded over here. These are the major projects that we are handling over here. That's an INSACOG, which is a genomic COVID genomic surveillance project, and the Genome India project, which is basically the human genome sequencing ethnic populations of India. I'll go into details of this. Now, as I said, that it is being developed in a modular fashion. The first module that we developed was the module to take up nucleotide data submission at IBDC. And for this, we have developed 2 data portals. One is known as INDA, that is Indian Nucleotide Data Archive. And the other one is Indian Nucleotide Data Archive Controlled Access. So here you can see that there is a difference. Their format and their intake formats of data are similar. They are identical. They differ only in how they provide accessibility to the data. So INDA is in real-time sync with all EMBL, NCBI, and DDBJ, that is GenBank and everything. They are in sync with these data repositories. So if you submit any data to INDA, any nucleotide sequence to INDA, you will automatically get accessions from INDA as well as EMBL, GenBank, and DDBJ. So now that problem that you needed to submit something on nucleotide sequence, you needed to submit it in GenBank, that is being solved. You can submit your data sets over here. And from here itself, you will get all the accession IDs which are required at the time of publication. That is a GenBank or DDBG or EMBIL IDs. So this solves the two purposes that we know the data is housed over here. And this is going by the international norms. All the formats are internationally accepted. And seamlessly, the data centers are integrated. This one, INDA-CA, it follows the same data formats, but here the data is not shared with any international repository. It stays in India. So this is where you can actually hold the data for a longer time. You can submit the data over here. And once you are ready over here, and then you can actually migrate it to INDA also, and then get your GenBank accession IDs. Or if you want to this data to remain non-accessible, especially in kind of, if the sensitive data, it will remain only on Indian servers. So this is the major first step that has been taken for the data for submission of the nucleotide data set, because nucleotide data becomes base of a lot of other data types also. So that's why that's the bulk of data that is being generated these days. Now, we are able to take any kind of nucleotide data, be it NGS data, genome level transcriptome assemblies, template-based submissions. All these different kind of data sets can be submitted. Any kind of study sample from, this is a plant sample checklist. You can have a whole lot of other different checklists where you can submit, you can think of any kind of a data type, even from depth of any ocean, that can be submitted. And we have formats for that kind of a submission. Metagenome also can be submitted, RNA-seq, everything. All of these can be submitted. And you can immediately get, if you want, you can immediately get INSDC, or that basically means GenBank submission IDs from here. What is the current states? We have already actually accumulated in the last 4 or 5 months only, more than 2 lakh submissions have already happened over here in INDA-CA. And in INDA also, the number of entries in INDA is less, but the size of these is quite big actually. And more data sets are coming in, more genome sequences are coming in. First plant genome that was submitted to INDA was the Brassica carinata genome assembly. And this is a submission that is for the first time anywhere. It has been done to INDA. And from here it has gone to GenBank and other data centers. Now, this is one mode of operation that I'll discuss now for the what the data center is doing to address issues at national level. So if you recall, there is a consortium of labs known as the INSACOG labs, which are basically involved in the genomic surveillance of the COVID-19 infection. So IBDC has become the de facto data portal for INSACOG, and all the data that is being submitted in these from across India are being submitted to IBDC now. This is the data portal over here. So if I just go back and show you this, if you click on this icon, actually, you will come up to this page. And this shows the real-time data submission from different labs across India. You can see that Government Doon Medical College, Dehradun, submitted 66 genome samples on 2nd of December this year. So, and as the data gets submitted, it gets reflected in this map from where the data is originating. And you can see that the most of the genomic surveillance is happening in the states of West Bengal and Kerala and Karnataka. So this gives a real-time idea of how genomic surveillance, and there are about 54 labs which are actually generating data and submitting sequences to up. And we have accumulated more than 2 lakh genome sequences over here. Now, besides just taking the data, we have also provided in InSACOG genome analytics services also. So what we are doing is whatever data is generated, whatever data is submitted to us, we are actually doing the variant calls, and you can see these are the distribution of the different COVID variants which are prevalent in India. And this is done in a daily fashion, and we give weekly reports to the InSACOG consortium. And you can see this graph over here. This is all available online, actually. You can just go on the tab and you can see this. And this is a weekly distribution of different variants. So it starts from first week of 2020, and then we are in the 41st week of 2022. This is the collection date. So right now we are in a little ahead, but the collection date would be running a little slow. And you can see that data analysis is done, and whatever call you see in the news where that XBB is coming up in a major is actually picked up from here. And you can see that XBB is currently is almost 50% of the entire kind of all type of variants which are found in India. This is the monthly distribution, the same thing. And similarly, we have other distributions also. You can go on the web and look at it. And other in the sense that what is happening in the northern part of India, southern part of India, that also is there, and you can take a look at that. Secondly, we have also handling a another major project, as I said, is the Genome India project, where they have now started to share the data. We have now 200 human genome samples from different states. You can see that these are the points from where these 200 samples are collected. And right now, more samples are being submitted. Now we have about 600 human genome samples, and the target is about 10,000 human genome samples, and slowly that will get accumulated and submitted over here. So besides this nucleotide data submission, other data portals are also in line, and they are being developed. The first one that will come across probably in a month's time is the Indian crop phenotype data phenome database, which is going to collect all phenotypic data sets which are being generated by several projects for phenotyping in different crops. So we have developed a portal for that. Similarly, the structural biology data for nucleic acids and proteins, structural biology data portal is also under construction, and probably in a couple of months, this will also come up as a major portal for IBDC. So this is how different portals are being developed for handling different kind of data types. And in years to come, I mean, this has just been over 1 year or now, 1.5 years now, and there has been an explosion of activities. We have been able to move very, very fast. Rather, during the lockdown period, if you recall, in last April, there was a lockdown in Delhi and Faridabad and adjoining areas. But that was the time that was, for us, it was the time of major activity where we were able to do, and we were actually able to make first submission in the month of, I think, July. So from within April to July, within the lockdown period, we were able to initiate the activities and establish the data center. The long-term idea is that the basic idea at IBDC is that it has to enable science in all research institutes, including universities and colleges. And it is not to infringe any benefit. That is, the data center will not infringe any benefit in the sense that if it is taking up the data, it's not going to it's not going to do any analysis on their own. It's not going to make a study of their own, but it is just a simple purpose that a data consolidation must happen. And if the research institute if the research group wants us to do some kind of analysis, they give us in written, and then we perform the analysis for them. So the basic idea is that IBDC will become a nucleation point for developing a kind of a biogrid of research labs which are connected to each other for data sharing and data analysis. And as well as what is most important is data standards and data analytic capacities. So we intend to inculcate and develop and bring at par all data centers, all research centers across lab for their capacity in data analytics and data sharing. So that a biogrid kind of a thing, a distributed biogrid, is something that we are working at. Now, we conduct regular workshops, online workshops. We even send our teams to places for orientation. We have, within few months, we have conducted more than 40 workshops. Every week, almost, we are doing a workshop over here. And we are very, very active in establishing one-to-one connect. That there are a lot of labs which were earlier not submitting any kind of data, but now they are submitting genome sequence data. So they get into some kind of issue. So we handhold them one by one and help them in their submissions, and even in data analysis also. So there's an extensive outreach program that we are implementing. And as I said, if you look at our website, our tagline is enabling science. So the basic idea is that we want to assist any lab which is interested in doing good science for data analytics. We go and send people and a group of scientists from our institute to other labs also to discuss different aspects of data submission and data analysis. This is the email ID which can be used by any one of you. You can write to us. We will immediately respond. The team is very responsive. You will get the response within the same day. And this is one thing that we are very, very concerned, and we are very precise about it, that our response have to be very good and very fast. And we try to solve the problem at the first. So this is the team. We have about 23, 24 people over here who are involved. They involve scientists. We have software developers. We have system managers and data curators. And they are all involved in developing and carrying out the activities of the data center. This is when on the day of the inauguration of the data center. So that's all from my side. I'd like to thank all the audience for their patience, and I would be happy to take any questions if there are. Thank you.
Thank you so much, sir. Now the